Just like you can turn the lights and other electronic things on and off, cells have the ability to regulate or control which genes turn on or off at any given time. They do this to respond to their environment. Both prokaryotes and eukaryotes can control which genes are expressed and which are not. They do this because it would be a waste of energy to make proteins that are not used. Prokaryotes, like bacteria, are simple organisms. Their genes are organized in groups that code for similar kinds of proteins. These groups of genes are contained in operons. Each operon is made of the genes that code for proteins. An operator, which works as the on-off switch. And a promoter, which is where the RNA polymerase attaches. There are also regulatory genes that are in a different section of the DNA that help decide if the genes in operon will be turned on or off. Eukaryotic cells are much more complex. Regulation can occur at any stage, from the start of transcription to the processing of a protein after translation. One way eukaryotes regulate gene expression is during transcription. RNA polymerase attaches to a promoter only with the help of special proteins. These proteins are called transcription factors. They bind to DNA and help turn genes on or off. Regulatory proteins called activators make it easier for RNA polymerase and transcription factors to bind to the promoter. Repressors do the opposite. They make it harder for RNA polymerase and transcription factors to bind to the promoter. Remember that DNA in cells is coiled into chromatin so that it fits inside the nucleus. Well, if the DNA is coiled really tightly, then it can't be transcribed. This can happen at the nucleosome, but the cell also has enzymes that make the chromatin either condense more or relax.